Just give me a minute. Okay. So ये सच में हो रहा है कि there is increased interaction. How do you prove that? So first what you've seen is that the microtubules and the dyneins seem to overlap more in one mutant. And this, if you can purify this, you know, simple thing. You have a cell, you see a lot of dyneins in the cytoplasm, you see some dyneins in the microtubule. If you can somehow purify the microtubules out of the cell, away from the rest of the cytoplasm, and now if you try to see how much of dynein is attached to the microtubule, then you can understand, or you can sort of try to have a quantitation, or semi-quantitation I should say, of how much these mutants interact with the microtubule. So it's something similar to how kinesins were identified. If you remember, kinesins identification was done by extracting the microtubules and then finding out what is exactly moving on the microtubules, because there were beads that were attached to microtubules and the beads were moving on microtubules. So if you now purify the microtubules, now you try to identify what was moving the beads. So what are the proteins there? Very similar thing. So what you do is you take the cell and there are here, there are here, how do I remove this? There are here a number of conditions that are shown on this. It's a huge number of plots, right? So amongst you who has not ever seen a Western blot? किसने आज तक वेस्टर्न ब्लॉट देखा नहीं है, जस्ट पढ़ा है? सभी ने देखा है? Then I will go ahead and explain this. So so let us look at each of these individual things. यहाँ पे क्या क्या है पहले. So MT stands for microtubule. तो किस लेन में माइक्रोट्यूब्यूल है और किस लेन में माइक्रोट्यूब्यूल नहीं, wait. MT is not microtubule. I think MT में क्या था? Yeah, MT or microtubule नहीं, was precipitated with or without paclitaxel stabilized microtubules. Okay, so this is, yes, it is without microtubules or with microtubules. So MT is microtubules and ATP तो आपको पता है, ATP is ATP. I is, you take the cell, you burst open the cell so that all the cellular contents come out, and that is your I, that is your input कि cell में क्या है. Everything that is in the cell will be there. Now if you burst open the cell, take out all its extracts and you spin it at very high speeds, then what is happening when you spin it? When you spin it, you are increasing the G-force on each of these molecules. So anything that is heavy now, any big protein complex, huge protein complexes, specifically those attached to, say, membranous components, they start to precipitate out. So this is a very easy technique to precipitate out cytoskeleton proteins, for example microtubules, actin. So age old technique. तो आप cell को burst कीजिए, क्योंकि these fibrous cytoskeleton are very often associated with the lipid cell membrane, etc. They tend to precipitate out. Whereas if the same things are not, so if you only have, say, actin subunits in the cytoplasm, or microtubule subunits not associated with this filamentous structure, then they will not precipitate out because they are not part of this huge network. Very easy method to isolate microtubules like tubulin structures or polymerized actin filaments. So they did that, they just spun it. Spin करने से क्या होता है? You will have a pellet that is all that network structure, and then you will have a supernatant. Supernatant में क्या होगा? Unpolymerized part. So you will have a polymerized part in the supernatant, polymerized part in the pellet and unpolymerized part in the supernatant. So this is what the indications here are. You have a microtubule, you've added microtubule or you've added ATP. These are the different conditions. You have the input, you have the supernatant, which is the unpolymerized part, pellet, which is the polymerized part. And this first blot indicates the wild type. B, C, D, E are the subsequent mutations of P1, P2, P3, P4. If we recapitulate, P1 और P3 में हमने देखा था माइक्रोट्यूब्यूल्स के साथ इंटरैक्शन ज्यादा है, right? Those mutants interact very heavily with microtubules. What do we see here? We see here that it just concentrate on these two lanes, these two and these two lanes. So this is in the absence of ATP. In the absence of ATP, this 380 band is for your dynein. It's a truncated dynein, remember? So it's a shorter dynein. You see, when you do not have ATP, most of your dynein is in the pellet. Most of the dynein. But when you add ATP, you see from the pellet it goes to the supernatant. That is, if you do not have ATP, most of the dynein seems to be very tightly attached to the microtubules. Microtubules are also in the pellet. Very tightly. But if you add ATP, a part of it just detaches from the microtubules and you can see it in the soluble fraction. So this is in the soluble fraction. This is for the wild type. If you mutate P1, in the absence of ATP, okay, it's still strongly binding to the microtubules. It's almost all in the pellet. But if you add ATP, you see the situation has not changed compared to this. It's nearly the same. This is P1. P2, you see, you kind of think of it as a ratio. Again, this is very similar to this case, not this case. P3, again, this is not as distinct change as this, neither is it a distinct change like this. And P4. So the most distinct change they found was that if you mutate P1 को हमने mutate किया, तो in the presence of ATP, the dynein does not seem to detach from microtubules. Rather, it is still very heavily associated with microtubules. What did they find? Most of the purified dynein, wild-type dynein, precipitated with microtubules in the absence of ATP. So when you don't add ATP, your wild-type dynein is heavily attached to microtubule. You add ATP, the wild-type dynein detaches. That's the first thing. The second thing they observed was that for the P1 and somewhat for the P3 mutants, majority of precipitated microtubules in the presence as well as absence of ATP. So whether or not there is ATP, if you mutate the P1 residue and somewhat the P3 residue, you see that majority of the microtubule is still attached, majority of the dynein is still attached to microtubule. It has not dissociated from the microtubule, which is exactly what they found here. For P1 and P3, most of the microtubule is actually yellow, which indicates that it's all throughout covered by these dynein mutants. Gives us very interesting feedback about the mechanism by which dynein moves on microtubules. So this is what we had seen in our last class, and we had tried to draw analogy with myosins in the way that myosin cycle में क्या होता है? If you think of the myosin cycle, you have when it is bound to ADP plus PI, that is a weak binding state on actin. PI is released, power stroke, strong binding. ADP is now released, ATP binds, and sort of it kind of detaches, right? ATP is hydrolyzed to ADP plus PI, again a weak interaction. That was myosins. Kinesins is also doing something very similar. So if you do not have an ATP bound to the kinesin head, kinesin head, Dynein head, because the dyneins have these 6 AAA plus ATPase ring. They bind to ATP. So if you now consider a situation where it is bound to ATP, then that situation shows us a state where the dynein is not able to bind to microtubule. So it is detached from microtubule. This is what we had seen the last time. तो यहाँ पे हम वही देख रहे हैं. आप ATP जब आप ATP नहीं डाल रहे हैं इस सिस्टम में, you have most of the dynein associated with the pellet fraction, that is most of it with microtubule. You add ATP and most of it goes in the soluble fraction, so it releases from microtubule, right? So okay, let's come to the next thing. So the next thing that they did was, because now they have all of these mutants and they know the affinity of each of these mutants for microtubule in absence and presence of ATP. The next thing is to test for ATPase activity. So they did that, and if you look at the ATPase activity, very carefully look at the y-axis. If you see the y-axis labels, you will see the y-axis labels are quite different. This one is wild type, normal. This is P1 mutant, the P1 mutant that sort of always kept tightly associated with microtubules. It's tightly associated with microtubules, but its ATPase activity is very, very low. You look at the y-axis here, compare it with the y-axis here for wild type. So very low, and it does not increase. It kind of, even if it picks up, it stagnates. That is, the ATPase activity is almost dead for the P1 mutant. P2 almost like wild type. P3 again kind of dead. इसमें ATPase activity है नहीं. P4 almost like wild type. Now, sometimes in biology we try to see if two things behave very similarly, say A and B behaves very similarly, and they are also working in the same pathway, then we try to see if दोनों को हम एक साथ डालते हैं, then does that add up? For example, P1 here is showing very high microtubule binding. P3 is also showing microtubule binding higher than wild type. And P1 is not showing ATPase activity. P3 is also not showing ATPase activity or very low ATPase activity. If now we combine P1 and P3, does it make it even worse? If that is the case, then you can say that P1 and P3 both contribute towards ATPase activity. However, if that is not the case, then you can say that one of these actually is the dominant ATPase, not the other. So that is what they did. They now mutated both P1 and P3, and they found that it is not additive. It behaves very much like P1, which indicates that P1 seems to be the dominant ATPase subdomain in dynein. तो ये जो हमने last time पढ़ा था, that P1, so last time we had said that this P1 subdomain has the dominant ATPase function. P3, if you disturb it, it somewhat affects dynein function, but P1 is the dominant. If you kill ATPase function of P1, you kill the dynein. So this shows how they found it. So you mutate that specific lysine residue and you study the individual tubulin binding, ATPase activity, and the third function is microtubule sliding function, because ultimately क्या हुआ? One is the dynein has to bind to microtubule. Second, it has to hydrolyze ATP. Third, it has to move. It has to walk on microtubule, right? If it is walking on microtubule, it could also be sliding microtubules. You remember kinesins? Kinesins where either the beads could be moving on the kinesin, or if you leave out the beads, you have fluorescently stained microtubules and you have not stuck them on a slide. You can also see that the microtubules are moving because of the motor function. जैसे ये motor चल रहा है, ये microtubule भी ऐसे slide कर रहा है. Something like this can also be envisioned for dynein. You have a dynein which is moving and the microtubules will be sliding past. And if you observe this under a microscope over certain time, you can calculate the rate of sliding of the microtubules. So that's what they did. What they found is this is the velocity in terms of wild type. P1 is dead. You mutate P1, you have a very high affinity for microtubule, doesn't have an ATPase function, and doesn't slide microtubules. So P1 mutated, so P1 is the AAA1 domain, subdomain of Dynein. You mutate that, you mutate its ATPase function, and you create a Dynein that attaches very tightly to microtubules, but does not cause any translocation. So it's a movement dead mutant. It does not move. P2 does not have that activity. P3, it has some sliding activity. As you could see here, it's not kind of completely dead. If you look, this is P1, P2, P3. Unlike this, it does have a little change in the levels of the dynein that is associated with the pellet versus dynein that is in the supernatant. Also, if you look here, this P3 mutant seems to have at least some associated ATPase activity, even if it is very low. But here you can see it has some amount of sliding activity, but very low compared to wild type. But it's not dead. It's definitely not dead like P1. And this is P4. So why is this study so important? Because this study is going to show you a few things. One is that this whole cycle of how the power stroke is generated in a dynein and what function ATP has in this whole thing. So you see, ATP associated dyneins, if you hydrolyze ATP to ADP plus PI, that is a state where it can bind to actin. That is a state where it can bind to actin. But if the PI is released, that is when it is creating this power stroke. And what we see is that this is a mutant that does not hydrolyze ATP. ATP hydrolysis is dead. ATP hydrolysis is dead, but it is able to very tightly attach to microtubules and it is unable to slide microtubules because ATP hydrolysis nahi hai. It doesn't undergo the conformational changes required for it to create that power stroke. So, okay, I'll not go into any further. Maybe we can take it in the next class. I don't know what the time is. Do we have any more time? We have about 5-6 minutes. So I will stop here for today's class. What is important in this class is that here is a very simple way to study how these motor proteins or what subdomain of the motor protein is important for ATPase function. Unlike the other motor proteins you have studied, tubulin, of course, tubulin you had two dimers, alpha and beta, and one of the dimer, the cleft is so guarded by the other dimer that the GTP hydrolysis does not occur. But for the others, you only had one single, so stoichiometry of binding of ATP or GTP was 1, right? Here for every dynein molecule, you could have multiple ATPs binding to it. So it was important to understand which part of dynein is actually responsible for ATP hydrolysis, actually responsible for creating this entire power stroke. So they did this mutation analysis, mutation scan. Before that, they had a pre-idea from the sequence, from a structural analysis that P1, so AA1, AA2, AA3, AA4, they are potential ATPase hydrolysis subdomains, not AA5 and AA6. So then they took out those residues that are required for binding and hydrolysis of ATP, mutated them one by one in each of these AA1, 2, 3, 4, and then studied the mutations individually to see which mutation can abolish ATPase function, which mutation can abolish microtubule sliding activity. So very simple paper. You can go through this paper. It will give you a preliminary idea of what are the techniques that are used in to study proteins in general, to study protein functions in general. Nothing to do specifically with microtubules. Very generally, how do you study a protein function? कोई भी protein function हो सकता है. आपके fermentation में कोई enzyme है, उसका भी function हो सकता है. How do you study it? So you need to look at one functional aspect, you need to look at a structural aspect, and how to do that. You can do a mutation and then study it. For mutation, you need to know the function, the structural aspect, कहाँ पे mutate करना है. इसका थोड़ा preliminary idea आप लोगों को मिल जाएगा. Okay? So specifically रत्नेश, ये आपके लिए कह रहे हैं क्योंकि आप इस बैच में अकेले बीटेक स्टूडेंट हैं और बहुत जूनियर हैं बाकियों से. तो आपके लिए कह रहे हैं कि आप ये पेपर को थोड़ा पढ़ लीजिए. If you have any difficulty, get back to me. With that, I will stop the class and I will take questions. Are there any questions? No? What I have taught today, is it clear or was it too difficult?
It was clear.
Okay, okay. All right. So I will send this paper today on to your backpack. Have a look at the article. As I said, it's a very simple paper. You will be able to understand it, but if there is any difficulty, please get back to me, because this is kind of how shall I say, as simple as it can get. इससे simple कोई paper आपको मिलेगा नहीं इस topic पे. Next time we will look at the Dynein function, but again थोड़ा सा कैसे लोगों ने किया है वो उस तरह उस एंगल से देखेंगे थोड़ा सा. ठीक है? So that is what we will take up in the next class. Sometime at the end of this week, I will send back all your answer scripts. And so what is the final conclusion? क्या करें हम? क्या हम 30% पे रख दें minor या 40% पे रखें?
30% is fine by me.
Okay, okay, thank you. So I will also send you your first assignment. It's a simple assignment. If anybody has any problem doing the assignment, do not hesitate to get back to me. As I said, मैं यहाँ पे आप लोगों को grades अच्छा देने के लिए ही बैठी हूँ. लेकिन अगर आप लोग खराब परफॉर्म करेंगे तो मेरे लिए grades देना बहुत मुश्किल हो जाएगा. I cannot justify that to myself. Okay. Thank you so much. Thank you, students. See you all tomorrow. And रत्नेश, आज क्लास के बाद प्लीज 5 मिनट आप वेट कीजिए, मुझे आपसे कुछ बात करनी है. I'll stop recording this. Sorry, I did not start recording initially. I just ये रिकॉर्ड हो रहा था ना. How do I stop recording it? Stop sharing. Stop recording. Okay.